So we just talked all about transcription, so now we know that we have our RNA has been completely transcribed from our DNA template in the nucleus. In bacterial cells, this doesn't happen this next step. That RNA is our mature RNA and it goes on to be translated. But in eukaryotic cells, there's another step between transcription and translation called mRNA processing that we need to talk about, and that's what 17.3 is about. So basically there's enzymes in the nucleus of the eukaryotic cells, obviously, because only eukaryotic cells have nuclei. There's enzymes in there that process the mRNA. They make, or pre-mRNA is what we call it because it's not ready yet. It's not our mature mRNA yet. And they make changes to the mRNA, to the pre-mRNA before it's ready to go out for translation. Dispatched to the cytoplasm for translation, as this says. And basically both ends of the pre-mRNA are modified, and usually some of the middle parts are cut out and rearranged a little bit. So those are the two things that happen here. Okay, so like I said, the pre-mRNA ends are always modified. The 5' end, as soon as it's made, so remember we add on to the 3' end, so the 5' end is just growing for a while off of the DNA template. And so as soon as it's ready, even though transcription is still happening, the 5' end is going to get a 5' cap. And what that is a modified form of guanine is put on at the end, of the 5' end, and okay, so it receives one modified guanine on at the end. It's called the 5' cap. At the 3' end, as soon as the pre-mRNA is done being transcribed, it receives the poly-A tail. Poly meaning many, A meaning adenine. So between 50 and 250 adenines are added on to the 3' end. These don't code for anything, obviously. They just act as the 3' cap, 3' end. And it is the, remember how at the end of transcription, the RNA polymerase reads the polyadenylation signal, AAU, AAA, and that's how it knows to stop transcription, and that's when it knows to add on the poly-A tail. So as soon as transcription is done, that's when the poly-A tail gets added to the 3' end, but once transcription starts and is still happening, that's when the 5' end receives the 5' cap of the guanine. And the reason for these modifications, because they always happen, are a couple of things. The first one is that it helps the mature mRNA, once the modifications are all done, helps it leave the nucleus. So they seem to allow it to pass through the nuclear pores, or however it's leaving, more easily to get to the cytoplasm for translation. So that's the first step, or the first reason. Second reason is that they help the mRNA from being broken down. So all throughout the cell, there's enzymes, hydrolytic enzymes. Remember, hydrolysis is when we break the bonds between monomers and we put in a water molecule. There's hydrolytic enzymes all throughout the cell, basically chomping on macromolecules and breaking them down. And so by adding on buffers on the ends, it helps prevent the important parts of the mRNA from getting broken down by the hydrolytic enzymes. And then lastly, it also helps the ribosome know what's going on when translation is about to happen. So when translation is about to happen at the ribosome, which is the next section, but when the ribosome needs to recognize the mRNA, and so it helps, these extra things help the ribosome recognize the mRNA and help it, help the ribosome attach to the 5' end of the mRNA. So it does attach at the 5' end, to the 5' cap. So that's the alteration of the mRNA ends, which happens all the time. So here's a pre-mRNA. So here is the 5' cap. Here's the poly-A tail. You can see there's a bajillion A's there. And here's our modified guanine. It has 3 phosphates. And then Here's the polyadenylation signal, AAUAA. And then you can see there's this 5' UTR and the 3' UTR, which are around the protein coding segment. So UTR means untranslated region. So when these guys get to the ribosome during translation, these sections right here, the 5' UTR and the 3' UTR, are not going to be translated. They're important for helping translation happen, but they don't actually go into the protein. Only this part right here, the protein coding segment, does go. And then there's something else, which is where we cut out middle parts of the DNA, of the mRNA, which is called RNA splicing. So splicing is when you cut it and cut something out and you seal it back together again. So most eukaryotic genes, and because of that, their RNA transcripts have long non-coding regions in the middle of them. And these are called intervening sequences or introns. Introns are in the way, they do not get made into a protein. And then all the regions that are important and will be expressed as a protein are exons, because they are going to be expressed. Exons are expressed, introns are in the way. And they will get translated into an amino acid sequence. So the splicing cuts out the introns and seals the ends back together so that we only have exons left, because that's all we want, just exons. So for example, here's our pre-mRNA, here's our 5' cap, our poly A tail, and then all this stuff is the RNA, but we only want the codons 1 through 30, 31 through 104, and 105 through 46. We don't want all these other intron things. They're in the way. We don't want them. They don't help us build our protein. So what happens is a bunch of enzymes come along, they cut out the section, and they seal the ends back together, and then we have our mature mRNA. So that is our splicing. And this is done by a molecule called a spliceosome. You don't need to know more details than that about it, but just know that the spliceosomes are what splice out the introns and seal the exons back together. So here's our intron of our pre-mRNA. The spliceosome comes along, cuts out the intron, and we're left with our mature mRNA. And our intron we don't need anymore. And that's all you need to know. Oh, just kidding. So people kind of say, what's the importance of introns? And the importance of introns is, they don't go into proteins, but they can help regulate gene expression, and we're going to talk more about that in a couple of chapters, but basically they help say when certain genes are going to be made into proteins and when not. For example, if you cut yourself and you're bleeding heavily, you're going to want proteins that help you stop bleeding, but you don't need those proteins all the time. So introns can help regulate things like that. And also, sometimes introns get cut out differently. So sometimes we might cut out a lot of introns from one gene, but we may also only cut out 1 or 2 introns from the same gene. So alternative RNA splicing is when you cut out different kinds of introns from the same gene, and you're going to get different gene products or different proteins. So that is why the textbook says that humans have the same number of genes as a nematode, which is a microscopic worm. And the reason why we have the same amount of genes, yet obviously we have a lot more diversity in our bodies, is because we do alternative gene splicing. So we can get a lot of different kinds of proteins or gene products from the same amount of information, because if you cut out this section of DNA, then you're going to get a different protein, or if you cut out this section, you're going to get a different protein. So alternative RNA splicing is really important. And that is it for that section.